For our project, we did a mitogenomic comparison of the cat family Felidae to show an evolutionary relationship. The biological family of cats, Felidae, is up of two subclasses, Felinae and Pantherae. Felinae is more commonly associated with smaller cats, like the domestic house cat and several kinds of lynxes. In contrast, Pantherae subclass is associated with larger cats, like the tigers, lions, and leopards. All cats are carnivores and they all have mitochondria, which is an organelle commonly known as the powerhouse of a cell. Mitochondria also contain their own kind of DNA, which is critical for energy conversion, but this DNA can also be used to determine the evolutionary relationship of cats. Recent research so far has been able to demonstrate evolutionary relationships among Felidae, but we wanted to explore that further to test the accuracy of the evolutionary history among various cat species.
Onto our hypothesis and objective. Although Felidae is classified into two subgroups, more data is needed explaining the reason for this classification and their evolutionary history. Our research aims to show an evolutionary understanding of Felidae species using whole mitochondria genome sequences, thus explaining their classification. Furthermore, if we blast mitochondrial genome sequences of these species and construct a phylogenetic tree in MEGA, we should see significant differences in evolutionary rates among the different groups of the family Felidae. Onto the methods portion of our project. First, GenBank was used to look up cat species. Complete mitochondrial genome sequences of 16 Felidae species and one outgroup, Canis lupus, which is the dog, were acquired using NCBI's BLAST tool. Mega software was then downloaded to build multiple sequence alignments of the Felidae species and Canis lupus. Once multiple sequence alignments were made, excessive sequences at the beginning and end of the genomes were cut to align perfectly, which will prevent similar species being grouped separately. When setting up the analysis, we used a bootstrap method of 1,000 replications and the distance correction method, such as the Kimura two-parameter model, to ensure the quality of the phylogenetic analysis. A phylogenetic tree was then constructed using the maximum likelihood model. Table 1 below displays the species along with their corresponding accession numbers from NCBI for the mitochondrial genome sequences incorporated in this study.
So after running our multiple sequence alignments through Mega, we produce a phylogenetic tree. And so here we see figure 1, which is a scaled version of the tree. And it is a bit easier to notice the relationships compared to figure 2. And groups of cats, they're color-coded based on the subfamily to which they belong to. So the blue belongs to the Pantherinae, or the big cats. And the green belongs to the Felinae, which are the smaller cats. And then we have the dog as the outgroup, and that is represented as a red color. And so when we interpret the phylogenetic tree, we want to look at the bootstrap values. And so in figure 2, we see that the bootstrap values are at the nodes of the phylogenetic tree. And in our analysis, we have a 100% bootstrap value in the group of Pantherinae, showing a low amount of sequence divergence. We also have a 100% bootstrap value in the Felinae group, showing very minimal sequence divergence. All of our bootstrap values are supported, especially ones above 70%, showing strong support of our data. The phylogenetic tree also has the scale bars that we can also look at, which represents the substitutions per site. The longer the distance, the higher the sequence of divergence. And so cats of the same species, like the different types of leopards within the same clade, they have short sequence divergence, which is indicated by short branches. You also see that with the different type of lions, since they are the same species, they have short branches indicating short sequence divergence. However, when we compare the branch lengths of the Felinae to the Panthera species, we see much longer branches, which is indicating that there's huge sequence divergence since they're the same different genus. And it's also true with the outgroup, which has the most extended branch of any other species. So our null hypothesis for this data is that there is no significant difference in evolutionary rates among different lineages in the phylogenetic tree. Our data came up with an R value of 0.9987 and a P value of less than 0.0001. Therefore, we reject the null hypothesis, meaning that there is evidence to suggest evolutionary rates are not independent among different groups. Our choice of using mitochondrial genome sequences offered many strengths. We say that the main strength is that it is maternally inherited in humans and animals, so it's a very good indicator when showing evolutionary relationship of closely related species. But there's also some limitations to take into consideration. So because it is only maternally inherited, it might not show a representative of both parental lineages. And also, we are only looking at a very small part of the genomic part of the body instead of like the whole entire nuclear genome. And so for future studies, it is best for researchers should use nuclear genomes that are highly conserved, which will enhance the reliability of phylogenetic trees. Our study advances functional genomics, showing evolutionary relationship between cat species with a detailed analysis of mitochondrial genomes. The analysis of these cats provides us a foundation for future research, giving us insight into genetic mechanisms concerning a diverse evolutionary relationship between different cat species. In conclusion, our results show significant differences in evolutionary rates among the lineages in the phylogenetic tree, and the correlation analysis using branch lengths indicates a strong positive correlation. These findings provide insights into the evolutionary history of cat species. And that concludes our presentation.